Good morning, students. In previous class, we have discussed about saturated fatty acids and polyunsaturated fatty acids. As we discussed in previous class that what are polyunsaturated fatty acids? The polyunsaturated fatty acids are which contains unsaturation at one or more stages or in more points, and they play protective role. When we talk about PUFA, then the first one name comes in our mind, that is omega 3 fatty acid. When we talk about omega 3 fatty acid, then there are lots of associated benefits are present. It lowers the triglyceride and cholesterol level. It is anti-inflammatory in nature. It beneficial for vision and brain function. It decreases skin inflammation. It inhibits platelet adhesion, increase insulin sensitivity. When we talk about omega 3 fatty acid, then it includes eicosapentaenoic acid and docosahexaenoic acid. Eicosapentaenoic acid is mostly present in fish liver oil, fish eggs, etc., which again get converted into docosapentaenoic acid, that is due to the presence of elongase enzyme. And when the docosapentaenoic acid is present over then delta 4 desaturase is responsible to convert docosapentaenoic acid into docosahexaenoic acid, which is needed for the development and function of brain. Basically, docosahexaenoic acid, that is DHA, is present in human milk, organic egg yolk, fish liver oils, fish eggs, liver, brain, and other organs. Now, omega 6 fatty acid. When we talk about omega 6 fatty acids, then these include linoleic fatty acid and arachidonic acid, and majorly the benefits are platelet aggregation, cardiovascular disease, and inflammation. It reduces the symptom of eczema and psoriasis and clear up different type of acne. When we talk about linoleic acid, the firstly linoleic acid gets converted into gamma linoleic acid, then again it is get converted into dihomo gamma linoleic acid. In next one step, again in one part it can be converted into arachidonic acid, which can be get converted into PGE2, prostaglandin 2, which is bad eicosanoids, whereas in if that dihomo gamma linoleic acid is getting converted into PGE1, then it is good eicosanoid in nature. Then when we talk about eicosanoids, then these compounds are derived from long chain polyonic fatty acid, where the carbon chain is with 20 carbons. The examples of eicosanoids are prostaglandins, prostaglandin, thromboxans, leukotrienes, lipoxins, etc. And now, what are the roles of eicosanoids? Regulation of blood pressure, blood clotting, essential for tissue growth, in asthma, in blood viscosity, in female conception, in increased male fertility, etc. Now, triglyceride. When we talk about triglyceride, the structure includes glycerol plus 3 fatty acids. That means there is one structure of glycerol and it is attached with 3 fatty acids. Those can be similar in nature or it can be in different nature. Now, what are the functions of triglyceride? Firstly, it is play important role in to give energy, and mostly the lipids, the energy which is coming from fat, it is around 9 kilocalories per gram. Forms stored energy in adipose tissue. It works as insulator and protector. It plays role in the carrier of fat soluble vitamins, and sensory properties of the food get increased due to triglycerides. Now, phospholipids, the main lipid constituent of cell membrane, where one glycerol, two fatty acid, and one phosphate group is attached over there. The functions of phospholipids are main lipid constituent of cell membrane, lipid transport as per a part of lipoprotein emulsifiers, and cell signaling process is done by phospholipids. Then there are a number of phospholipids which are given. Now, sterols. Sterols are the compounds contain cyclic steroid nucleus, namely cyclopentanophenoethrin ring. Cholesterol. The best non-steroid which can be produced by the body, and when we are going to consume it, the outside sources of fat, then it is getting to convert into cholesterol because of its association with atherosclerosis and heart disease. It is the known to steroid. It contains same steroid nucleus exclusively present in animals. The functions of cholesterol is bile acid production, then in the production of cell hormones, then it is also essential for the adrenaline hormone. It is essential for vitamin D and cardioglyceride. Now, glycolipids. First, when we talking about cholesterol, then when we talk about animal sources, in animal sources, it is present in the as such form, and if we are going to consume it from the dietary sources, then it again create problem and increase the cholesterol level in body. Whereas if we skip cholesterol consumption after that, or the consumption of the fat, if we skip that, after that it will be present in body because body is able to produce cholesterol itself. Glycolipids, it is widely distributed in the body, and the combination is fatty acids, sphingosine, and carbohydrates are attached over there. It also known as glycosphingolipids. It presents in cell membrane and nervous tissue. Then lipoprotein. Lipoproteins are the macromolecular complex of lipids as well as protein. Apolipoprotein. Apolipoprotein, ApoA1, that is present in liver and small intestine. ApoA2, that is present in liver. ApoA4 is present in small intestine, and ApoA5 is present in liver, which direct functional role is unknown, but it regulates triglyceride levels. Then chylomicron. It is synthesized in intestine. It transports endogenous triglyceride, and hydrophobic cores are there because the triglyceride are present in 93%, whereas cholesterol esters are present at 1%. This is the structure. This is the structure of high density lipoprotein. Now, the effect of various type of fat on blood lipid levels. When we talk about saturated fat, then that increase total cholesterol level and increase low density lipoprotein cholesterol level. When we talk about polyunsaturated fat, then it decreases total cholesterol level, decreases LDL cholesterol level, and increases HDL cholesterol level. When we talk about monounsaturated fatty acids, then decreases total cholesterol, decreases LDL level, and increases HDL level. When we talk about omega-3 fat, it decreases total cholesterol, decreases LDL level, increases HDL level, and decreases serum triglyceride level. Trans fats, it increase total cholesterol and increase LDL levels. Now, what are the advantages of lipids? It plays important role in our body. It provides energy. It helps in the absorption of fat soluble vitamins especially, and few are essential fatty acids for body, just like your EPA, DHA, linoleic acid, etc. What are the disadvantages of lipid? It raises your susceptibility to heart disease. It leads to the weight gain, then make you insulin resistant also, and it can increase the risk of cancer. Now these are the applications of lipids. Now, digestion and absorption of lipids. Since most of our digestive enzymes are water based, how does the body break down fat and make it available for the various function it must perform in the human body? When we talk about the digestion, it is the breakdown of large molecule of food molecules into small water soluble food molecules so that they can be absorbed in the bloodstream. Digestion is a form of catabolism that is often derived into two processes, that is mechanical and chemical digestion. Mechanical digestion includes the physical breakdown of the large pieces of food into smaller pieces, whereas chemical digestion includes enzymatic breakdown of food into small molecules and get utilized by the body. Foods such as meat, dairy products, seeds, nuts, and oil contain dietary fat. Fat is a common example of lipid. Fat does not like water. It tends to clump together and form large droplets as it moves through your digestive system. By the time fat reaches your small intestine, it has not been digested at all. So dietary fat is get digested in small intestine. When we talk about the major dietary lipids are triacylglycerol, cholesterol, phospholipids, etc. It includes, when we talk about the digestion and absorption of lipids in body, then it includes number of steps. Firstly, digestion in mouth. A very amount of digestion of lipid takes place in mouth due to the presence of lingual lipase. The lingual lipase enzyme is present in mouth in saliva, and where the optimum pH is 2.5 to 5. Its action continues in stomach also. Now, when the lipids passes through the esophagus, of course, the retention time is very less, so no digestion can be occur. After that, in the digestion part occur in stomach, then gastric lipase is present over there, and which is the enzyme which works for the digestion of lipid in stomach. It has an optimum pH of 5.4, and which is secreted by chief cells. Up to 30% of digestion of triacylglycerol is occur over there. Then the breakup of fat globules into much smaller emulsion droplets. The emulsion droplets are where digestion occur, and emulsification greatly increases the surface area where water soluble lipase can work to digest THG. The bile salts break up and coat the fat to form much finer droplets. So the role of emulsification is done by the bile juice. Bile, having the pH 7.7, entering the duodenum, neutralize the acid chyme from the stomach and provide favorable pH for the action of pancreatic lipase. When we talk about the lipolytic enzyme in intestine, then these are pancreatic lipase, colipase, cholesterol esterase, and phospholipase. In the small intestine, as pancreatic lipase enters the small intestine, it breaks down the fat into free fatty acids and monoglycerides. Now, the question is raising, how will the fat pass through the watery layer of mucus that coats the absorption lining of the digestive tract? So at this time, the bile play important role because there are bile salts are present in bile juice, and the bile salt works as it makes a coating around the fat molecule. The small droplets of fat get entrapped in the bile salt layering, and when the bile salt layer the whole droplet, then it will be able to pass the watery membrane. Micelle. After digestion, monoglyceride and fatty acid associate with bile salts and phospholipids to form micelle. Micelle are about 200 times smaller than emulsion drops. Micelles are necessary because they transport the poorly soluble monoglycerides and fatty acids to the surface of the enterocyte where they can be absorbed. And these micelles allow efficient transportation to the intestinal microvillas. This is the whole process where large lipid droplets are present over there. Firstly, they are getting converted into small droplets, and then it get mixed with the bile salts, where bile salts layer up the molecules, and it is going to become micelles. Now this micelle is going to work by the pancreatic lipase, and the fatty acid and monoglyceride will be get released, and after that they will be able to enter into mucosal cell. Now these are the associated parts, the lipids which contain in food, their application, self assembly and biology. Thank you.